All right, chapter 21 is going to be divided into two parts. It's all about the evolution of genes and genomes. In the first part, we'll talk about some of the mechanisms that can cause the evolution of genomes, help drive that evolutionary change, and talk a little bit about the ways that biologists study such change. And then in the second half, we'll discuss the fact that even just changes in gene expression patterns can often have very dramatic effects on evolutionary development, and also give you some practical applications for studying this field called molecular evolution. All right, so as you're hopefully aware, the genome is just defined as all the DNA in a cell, so all the genes plus the non coding and regulatory DNA. And also keep in mind some genomes, primarily those of viruses, can be made of RNA as well. Um, and so if we're thinking of changes to the genome, of course, that's going to be driven by mutation, which are, typically either caused by external factors like mutagens or just by mistakes in the process of DNA replication. And no matter how it happens, without mutation, you cannot have evolution. So you've got to have some genetic variation provided for by mutations in order for evolution to occur. And as we've uncovered these genome sequences and taken sort of a deep dive into studying genomes, this has opened up this new field called molecular evolution, which is studying molecular variation in gene sequences and protein amino acid sequences, and using that to try to reconstruct not just the evolutionary history of the genes themselves, but it could also be applied to studying the evolutionary history of whole groups of organisms. So, as we've said, right, genes and genomes are going to evolve by mutation, changes in the DNA sequence that are typically referred to as nucleotide substitutions. So, we're substituting one nitrogenous base for another, an A for a G or an A for a T, whatever the case might be. And anytime you change the sequence of a gene, that may also potentially change the sequence of the amino acids that are coded for in the protein. And if we change amino acids, that may alter the function of that protein. And typically, if we change amino acids in a protein, it's very likely that that may disable that protein. But on certain rare occasions, you may actually gain some new function. And that's really where things start to get interesting in terms of evolution. So, if you are studying molecular evolution, as I said, you're really focused on these nucleotide substitutions or amino acid substitutions. And as I'll show you on the next slide, really the basis of this kind of study is aligning and comparing sequences. And the idea is that the longer that two sequences have been evolving separately, the more different they're likely to be. Um, so, it's tempting to think of this as just, biologists sitting in front of a computer just pouring through, existing genome and DNA sequences. But I should point out, increasingly, evolutionary biologists and even those studying molecular evolution are able to use experimentation. In particular, this is going to be using single-celled organisms or viruses that have very short generation time. So biologists could actually induce mutations and observe not only the evolution of these populations of single-celled organisms or viruses, but observe the evolution of the genes themselves. But that being said, a lot of this does come back to comparing sequences and doing what are called sequence alignments, which is this technique for trying to get essentially a visual comparison between two, in this case, amino acid sequences, or more than two potentially. So let's just start with two. Let's say you wanted to do an alignment of sequence one and sequence two here. So we've got these short portions of a protein, these amino acid sequences here. And just at first glance, if we just start by aligning them directly, first amino acid, second amino acid, third, and doing the comparison that way, there's not a whole lot of similarity there, right? So the first amino acid is the same, but the second's different, the third's different, fourth is the same, fifth is different, so on. But I wanted to point out one very common technique that's used when doing these alignments is to introduce what are called gaps. So here we see if we just introduce this one gap in sequence two and do the alignment, now all of a sudden everything lines up much better, right? And we can see there's maybe a lot more similarity between these two sequences than we realized. Maybe somewhere along the line, this amino acid sequence from organism 2 actually lost an amino acid. And if we just account for that loss now, we see, yeah, they are in fact, very identical across their whole length. And of course, as I said, you could do this for many, many sequences at the same time. A lot of times this is being carried out by computer programs that will, search for potential locations to insert gaps to create the best alignments. So here's an example of a large comparison. This is the amino acid sequence of the cytochrome C protein. You may remember this was one of the key components of the electron transport chain that's important for cellular respiration. So basically any organism that carries out cellular respiration and has mitochondria is going to have a cytochrome C gene and cytochrome C protein. And this kind of alignment can be very useful if you're a researcher studying this particular protein, because when we do this comparison across lots of different types of organisms, we may be able to identify certain parts of the protein, certain amino acids that just never change, right? They're very consistent in a particular position in that protein, no matter what organism you're looking at. And if you see those kinds of amino acids that just never change, that would argue that having that specific amino acid and that specific position in the protein is probably very important, and it gives you some key information about what makes that protein tick, so to speak. And I should point out, I know we've already talked about mutations and some of the terms that go along with that, but the nomenclature is a little bit different in the field of molecular evolution. Um, so they will often refer to what they call synonymous versus non-synonymous substitutions. And these are amino acid substitutions. And you can think of synonymous substitutions as equivalent to a silent mutation or a silent substitution, right? Synonyms would be two different words that basically have the same meaning. So we can see in panel A here, we can actually have a change in the DNA sequence that doesn't alter the amino acid that's coded for by a particular codon. Remember, in many cases in the genetic code, if you change the third letter in the codon, so in this case UUA was changed to UUG, right? Both of those codons code for the same amino acid, leucine in this case. So we would consider those essentially synonymous codons. We had a synonymous substitution, and that's going to likely be silent, right? It's probably not going to have any effect on the protein. Whereas a non-synonymous substitution is going to be one that does change the amino acid. So previously we had referred to these as like a missense mutation, where we, for example, change a leucine into a phenylalanine, or a nonsense mutation where we change a amino acid encoding codon into a stop codon. So something along those lines. And again, as we discussed previously, these sort of non-synonymous substitutions are usually going to be harmful to the organism, but on very rare occasions, the organism or the protein may derive some benefit, may gain some new function. So a lot of what you'll see in this field of studying molecular evolution is, again, doing these alignments, looking at these different types of substitutions, and in particular, there's often a focus on the rate of synonymous versus non-synonymous substitutions, because that rate can give you some indication of how fast any particular gene is evolving. So, as we've studied the evolution of genomes, there's a couple of things that have been very clear. Um, First and foremost is that genomes are definitely changing over time, and they tend to be growing larger over time as well. So, if we look at just genome size across the many, many genomes that we've sequenced, we see there's a lot of variety out there. And there is some correlation between genome size and organism complexity. It's not a perfect correlation, but in general, we see more complex organisms tend to have larger genomes. So, as I said, it's not a perfect correlation, but in general, we do see that trend. And researchers have observed that a lot of the variation in genome size tends to be in the non coding DNA in particular. So, DNA that does not code for genes. So, just going back to that trend of increasing genome size, we can see that illustrated here, where we're looking at the number of genes, which is a pretty good indication of genome size. So this is number of genes times 1,000 here on the x-axis. And you can see we've got different groups of organisms. So we start up here with prokaryotes, right? Relatively small number of genes. We see that number increase as we get into some single-celled eukaryotes. And then we've got our multicellular eukaryotes, plants, fungi, animals, having the most genes of all, right? But as I said, it's not a perfect correlation in terms of complexity and genome size or number of genes, because if we look at this particular chart, like rice has the most genes and is arguably not the most complex organism on this list, considering we've got humans on here as well. But some of this difference just comes down to, especially in plants, chromosome number. So you may remember the fact that plants can tolerate polyploidy, having multiple copies of chromosomes very well. And I'm not sure off the top of my head what the ploidy number is of rice, but I'm certain it's pretty high. So because there's more copies of each chromosome, that sort of artificially inflates the number of genes. And again, we see something similar just looking at straight genome size here, right? Instead of just focusing on the number of genes specifically. But here we've got genome size on the x-axis, and then on the y-axis we've got the percent of the genome that actually codes for genes. And what you see in prokaryotes is you have a small number of genes, but almost 100% of the genome is genes, meaning there's basically not any or very little non-coding DNA. And then, we get some single-celled eukaryotes like yeast, have more genes than the prokaryotes, but also more non-coding DNA. And then as we get down here, we're into our multicellular eukaryotes, where we get, in many cases, a very small percentage of the genome is actually genes, but the genome size overall is much larger. So again, that's suggesting that as genomes grow, we're typically getting not only more genes, but a lot more non-coding DNA as well. And we know some of that consists of like repetitive DNA sequences and things like the transposable elements that we talked about in previous chapters. Remember, those were those DNA sequences that could copy themselves and reinsert. All right, so if genomes are arguably growing over evolutionary time, because all the evidence suggests life on Earth began in the form of prokaryotes with these small genomes, small number of genes, what are these mechanisms that are increasing genome size? And there's really two that I want to talk about. One is what's called lateral gene transfer, and the second is just called gene duplication. So, what does this mean? What is lateral gene transfer? Well, as that name implies, that's referring to the transfer of genes from one organism to another, sort of horizontally. Usually, when we think gene transfer, we're thinking parent to child. That would be a vertical transfer of genes from one generation to the next. But here we're talking horizontal from one species or one organisms to another. And this is, somewhat rare in eukaryotes. Um, one mechanism for lateral gene transfer in eukaryotes would be virus infection, because viruses can sometimes pick up a gene from a host and then deliver that into the genome of a newly infected individual. Um, but in prokaryotes and bacteria, um, they can actually, in many cases, just soak up DNA from their environment. So they're sort of like DNA sponges, and then incorporate some of those DNA fragments into their genome. Um, so that is going to result in pretty rapid and widespread lateral gene transfer in the prokaryotic world. Another mechanism that could occur in eukaryotes is hybridization, right, between species or subspecies, might be considered a form of lateral gene transfer as well. And so we see there can be some big advantages for organisms in terms of obtaining DNA through lateral gene transfer, particularly in the bacterial world. A lot of the antibiotic resistance that's been occurring in over the last couple of decades, we believe to be due to lateral gene transfer. So some of the genes that confer antibiotic resistance getting picked up from the environment from one bacterial species to another. Then the second big mechanism was what's called gene duplication, which is literally, as that name implies, just making more copies of an existing gene in a genome. And we'll talk about how that could happen here in a minute. But we know it happens with some regularity. And anytime you have a gene that becomes duplicated in a genome, so let's say you had one copy of gene X, it gets duplicated, now you have two copies. There's a few possible things that could happen. So one is the new copy may just retain the original function, and because we now have two copies of that gene, we're basically, that organism or cell is just making twice as much of the protein that's coded for by that gene. And that may or may not have any observable effect on the organism. A second possibility is that the new copy of the gene, or one of the copies, keeps the original function. The protein is still performing the same job, but the expression pattern changes, so that maybe the new copy is just being expressed in certain tissues, certain parts of the body, or maybe at some very specific time during embryonic development, or some brief period during that organism's life. So we sort of see some specialization in terms of the expression of that particular copy of the gene. A third possibility is that one of the two copies basically picks up mutations and becomes non-functional. It becomes what's usually referred to as a pseudogene, because if you look at the sequence of the genome, you see what looks like a copy of this gene, but it turns out, because of these mutations, that copy's not really doing anything biologically speaking. And then the last possibility is that one of the two copies accumulates some mutations, some substitutions that actually give it a new function. So now we've got something new and unique happening, biologically speaking. And anytime there's a gene duplication event, any one of these four things could potentially happen. It just depends on really random chance in terms of how it plays out. But we know gene duplication events happen with some regularity, and over time, if you have multiple gene duplication events in the evolutionary history of an organism, you end up with what evolutionary biologists call gene families. So, for example, here we're seeing an illustration of what's called the globin gene family. So, this would include like the alpha, beta globin genes that are used to produce hemoglobin, right? Carries oxygen in our bloodstream. Also includes sort of a more primitive version of hemoglobin called myoglobin, which stores oxygen in muscles. But we can see here in the human genome, there's multiple versions of these different globin genes, and it's believed that these have happened through multiple gene duplication events over time. So each one of these branch points here would represent a different gene duplication event. And if we zoom in and look at the function of these different genes, we really see all four of the different things that we were talking about here on the previous slide happening. So some of these globin genes seem to still have the original function. Basically, myoglobin is thought to be the most primitive of these, the most ancestral form, and we're still seeing it carry out what was believed to be its original function. Um, we see some of these just being expressed in very specific tissues or at specific times of development. We see some of these that are pseudogenes that sort of look like globin genes but don't actually seem to produce any functional protein. And then we've seen some of these develop new functions because it's believed we started with just a myoglobin or a myoglobin ancestor, and now we have alpha and beta hemoglobins that are doing something sort of unique and slightly different than that myoglobin ancestor was. So that's pretty typical. We can really see these gene families in pretty much any genome that we look at. All right, so lastly, to finish up this first part, how does gene duplication occur? We talked about some mechanisms for lateral gene transfer, um, bacteria soaking up DNA from their environment, viral infections, hybridization, but how do we get gene duplication events? And more often than not, this is going to happen due to unequal crossing over that occurs during meiosis. So if you remember, during prophase 1 of meiosis, the homologous chromosomes pair up and we get these crossover events where they exchange some DNA. And typically, when the homologous chromosomes align, they should align basically perfectly with each other. So when these crossovers do occur, they're swapping equal and complementary portions of the chromosome. But occasionally they will not line up correctly, as you see here on the left, right? They're not perfectly aligned. And now if a crossover event does happen, we'll end up with a situation like you see here on the right, where now you're going to have some chromatids that are missing information, like chromatid number 2 here, and some like chromatid number 3, where we've duplicated this particular region, this particular gene C in this case, right? So crossing over following uneven alignment of homologous chromosomes during meiosis is going to give us these duplication events. All right, I will continue this in part 2.